Welcome to the Neurostimulation Podcast. I'm Michael Passmore, Clinical Associate Professor in the Department of Psychiatry at the University of British Columbia in beautiful Vancouver, Canada. The Neurostimulation Podcast is all about bringing cutting-edge neuroscience education to you free of charge. Each week, we're going to explore the fascinating world of neuroscience in general and neurostimulation in particular. We're going to look at how it works, what are the latest research breakthroughs, and most importantly, how is that research being translated into real-world treatments that can improve health and well-being. So whether you're a healthcare professional, a student, or just someone who's curious about how our brains work and what we can do to help them work better, this podcast is for you. My mission is to make this science accessible, inspiring, and relevant to your life. As part of our commitment to evidence-based analysis, each episode will include an advanced critical appraisal of the study that we're talking about. If you're not interested in this deeper dive into the methodology and study analysis, feel free to skip ahead on the timeline to focus on other aspects of the discussion that you might be more interested in. This podcast is also separate from my clinical and academic roles, and it's part of my personal effort to bring neuroscience education to the general public. Accordingly, I'd like to emphasize that the information shared in this podcast is intended for educational purposes only, and is not intended as medical advice or a substitute for professional medical guidance. Always consult with your healthcare provider to discuss your specific health needs and treatment options. By listening to or watching this podcast, you acknowledge and agree that any decisions related to your health are your own responsibility and should be made in consultation with a qualified medical professional. This episode is presented by ZipStim Neurostimulation. Full disclosure, ZipStim is the neurostimulation clinic that I own and operate. You can find out more about our clinical neurostimulation programs at zipstim.com. That's ZIPSTIM.com. Have you ever wondered how scientists can target specific areas of the brain to study or even change using neurostimulation? How it works, all non-invasively? That's what we're talking about today. A cutting-edge technology called Transcranial Temporal Interference Stimulation, or tTIS. It's the focus of a fascinating study exploring how this method can affect motor learning. This particular study, published in the journal Nature Human Behaviour, explores how our brains learn from reinforcement, those little good job signals that we get when we succeed at something, and how tTIS can either enhance or disrupt this learning. The researchers focused on the striatum, which plays a key role in reinforcement learning and motor control. They wanted to see if tweaking the activity in the striatum with tTIS could shed light on how these processes work, and maybe even pave the way for new treatments for conditions like Parkinson's disease or ADHD. So what is tTIS? Let's do a basic overview. Imagine two pairs of electrodes, so 4 electrodes total, placed on the scalp strategically. These electrodes send tiny, safe electrical currents into the brain. But here's the interesting part. Each pair of electrodes generates a high-frequency current that's too fast for the local brain cells to react to directly. But when the currents meet deeper in the brain, they create a slower beat frequency that can actually influence the targeted neurons. Think of it like tuning 2 instruments to create a perfect harmony, except here the harmony is a specific frequency that targets the neurons in a deep brain region like the striatum. Let's talk for a minute about the bioengineering behind tTIS. How does this work biologically? Remember, neurons communicate through electrochemical signals, and their activity can change depending on the rhythm or oscillations of those signals. So by using tTIS, the researchers can deliver a specific frequency of energy to modulate the brain's natural rhythms. In this study, they tested 80 Hz, which is a high-frequency rhythm that's linked to reinforcement learning, and they compared it with a frequency of 20 Hz, which is associated with sensory motor functions. So by altering those rhythms, they essentially tuned the brain, kind of like adjusting a radio to a given radio station, in order to study the effects of that tTIS. The tTIS can reach deeper brain structures like the striatum, which other non-invasive neurostimulation techniques like transcranial magnetic stimulation struggles to do. This is why tTIS is so exciting. It combines precision with non-invasiveness. This particular study is not just about cool technology. It's also about understanding how our brains learn and adapt. By targeting the striatum, the researchers are trying to answer a big question. How do specific brain circuits help us improve our skills? And can we manipulate those circuits to help people who struggle with motor learning, like after a stroke, in Parkinson's disease, or other conditions? Have you ever tried learning a new skill, maybe playing an instrument, or if you think back to as a kid when you were learning to ride a bike? Remember when you felt that rush when you finally got it right? That's reinforcement learning in action. It's your brain's way of rewarding you for success, making it more likely that you'll do the same thing again next time. But how does this happen? And what does the striatum have to do with it? Reinforcement learning is like your brain's built-in feedback system. Think of it like a coach that's constantly saying, yes, do more of that, or no, try something else. When you succeed at something, your brain releases a little bit of dopamine, a feel-good chemical that strengthens the connection between your actions and the desired effect or reward. This process makes it more likely that you'll repeat the behavior. For example, imagine that you're learning to play a song on the piano. Every time you hit the right note, your brain reinforces that action, helping you remember what worked. Over time, you get better and better, thanks to this positive feedback loop. Now let's zoom in on the brain. The striatum is part of a group of deep brain structures called the basal ganglia. It's kind of like the brain's scorekeeper for reinforcement learning. It keeps track of actions and their outcomes, helping you to figure out what works and what doesn't work. Here's how it works. When you get positive feedback, like a reward for hitting the right piano key, the striatum activates and strengthens the neural connections that are responsible for that action. It's also tightly connected to other brain regions, like the motor cortex, which controls movement, and the prefrontal cortex, which handles planning and decision-making. Together, these areas form a team that help you learn and adapt. Reinforcement learning isn't just for skills like playing piano or riding a bike. It's happening all the time. It helps you learn everything from tying your shoes to making better choices at work. When you try something new and it works, the striatum helps store that information, making it easier next time. But here's the catch. When this system doesn't work properly, it can lead to big problems. For example, people with Parkinson's disease often struggle with motor learning because the connections between the striatum and these other important brain areas are disrupted. Understanding reinforcement learning can therefore help us develop better treatments for these kinds of conditions. Now I'd like to introduce you to someone who helps me to bring neuroscience education to life, literally. This here is Yorick, my trusty brain model.
How long will a man lay the earth ere he rot?
Oh, faith, if it be not rotten before he dies, some 8 year, 9 year. Here's a skull now. Lying you in the earth some 23 years.
Whose was it?
A horse and mad fell as it was. He poured a flagon of Rhenish on my head once. Whose do you think it was?
Nay, I know not.
The same skull, sir. What, sir? Yorick skull. The King's Jester.
This?
In that.
Let me see. Alas, poor Yorick.
I knew him, Horatio. A fellow of infinite jest, of most excellent fancy.
He's not quite as chatty as Shakespeare's Yorick from Hamlet. And luckily for us, he doesn't mind when we poke around in areas like, for example, his striatum. All right, Yorick, let's show us what you got. So you can see this is what we call a sagittal, mid-sagittal view here. So the striatum is going to be located deep inside the brain. You can't really see it here. It's going to be underneath this area deeper. But you can think about it like the brain's air traffic controller. It's important for making decisions based on rewards and feedback. Kind of like Yorick's little personal GPS system for learning what works and what doesn't. The striatum is divided into two main parts, the caudate nucleus and the putamen. If Yorick had a sweet tooth, these areas would be lighting up when he spotted a donut. These regions are going to help him learn from his choices, like whether reaching for that donut is a good idea or a bad one. For Yorick, it's always a good idea, but I digress. In our study today, tTIS focused on the striatum, which is why we're giving Yorick's striatum a little extra attention. By tweaking the activity here, the researchers were able to disrupt how the brain learns from rewards, kind of like reprogramming Yorick's GPS and suddenly sending him to the wrong destination. Very inconvenient if you're trying to master a skill. Thank you, Yorick, for lending your brains to this discussion. No skullduggery here, haha, just pure science. Now let's see how all of this ties into the results of the study. So why focus in on the striatum in this study? Well, the researchers knew that the striatum is crucial for reinforcement learning, but they wanted to dig deeper. By using tTIS to directly target this area, they could test how changing its activity affected learning. This approach helps us to understand not just how the striatum works, but also how we might use brain stimulation to improve learning in people who need it the most. So how do you test whether brain stimulation can change how we learn? Well, you start with a well-designed experiment and a bit of technological wizardry. Let's explore how the researchers used tTIS to uncover the role of the striatum in reinforcement learning. This study recruited 24 healthy young adults. The goal was to figure out how stimulating the striatum with different frequencies of tTIS would affect their ability to learn a motor task through reinforcement. It's kind of like playing a video game where you have feedback, like, say, a flashing green light, for doing the task correctly. Here's the setup. Participants were asked to perform what's called a force tracking task. They squeezed a hand grip device to match a moving target on a screen. Think of it as kind of like a high-tech version of playing that old game Operation, where precision of movement is everything. While participants were performing the task, the researchers applied tTIS to their brains using those two pairs of electrodes that we talked about, placed strategically on their heads. These electrodes sent high-frequency electrical currents deep into the brains of the participants to target the striatum. They tested 3 conditions. First, 80 Hz stimulation. This targeted high gamma oscillations, which are thought to play a role in reinforcement learning. They also tested 20 Hz stimulation, which is aimed at beta oscillations linked to sensory motor functions. And then they had a sham stimulation, which is basically a placebo, where no actual stimulation occurred. This careful study design allowed the researchers to compare the effects of different frequencies and rule out placebo effects. In order to test how reinforcement influences learning, participants completed 2 types of training. The first type was called reinforcement training. Participants got real-time feedback, like a green light, when they performed the task correctly. The second type was called random feedback. Here, feedback was randomized giving no useful information about performance. By comparing these two conditions, the researchers could isolate how reinforcement and the tTIS stimulation affected learning. In terms of tracking performance, the key measure was how well participants improved over time. The researchers tracked their performance during training and then tested them again afterwards without feedback to see how much they retained. It's like practicing a skill and then testing how well you remember it the next day. This study design is a gold standard for studying cause and effect. By combining real-time brain stimulation with a controlled task, the researchers could pinpoint how tTIS changed brain activity and its impact on learning. It's like running a highly controlled experiment on a human brain in real-time action. All right, now that we know how the experiment was set up, it's time for the big question. What did they discover? Did tTIS really affect how people learned? And if so, what does that mean for understanding the brain? Here's what the researchers found. When participants received 80 Hz tTIS during reinforcement learning, their ability to learn from feedback was disrupted. In other words, the stimulation interfered with the brain's natural ability to use those good job signals to improve performance. But when participants got either the 20 Hz stimulation or the sham treatment, their learning stayed intact. It would have been kind of like trying to practice free throws in basketball, but having someone constantly distract you with a loud noise. Except here, the distraction was actually happening inside the participant's brain. So why does the frequency of the tTIS matter? Well, these researchers believe that it comes down to how the brain's rhythms work. High gamma oscillations around 80 Hz in the striatum are thought to play a key role in reinforcement learning. So by disrupting these natural rhythms with tTIS at the same frequency, the brain couldn't process those reinforcement learning signals effectively. On the other hand, the beta oscillations at 20 Hz, which typically are more involved with sensory motor functions, didn't interfere with reinforcement learning in this same way. This shows how specific brain rhythms are tied to different types of learning. One of the most interesting findings was that the 80 Hz tTIS specifically affected activity in the striatum without impacting other brain areas. This precision is what makes tTIS so exciting. It's like having a laser-guided tool to non-invasively study deep brain regions, something that's hard to achieve with other neurostimulation techniques like transcranial magnetic stimulation. What about retention? This study didn't just look at the immediate performance. It also tested how well participants retained what they learned after training. And guess what? The disruption caused by 80 Hz tTIS carried over. Even when participants were tested later without feedback, those who received the 80 Hz stimulation struggled to perform as well as the others. This suggests that reinforcement learning doesn't just help in the moment, it also strengthens memory for future use. So what does all this mean? It's a major step forward in understanding how specific brain circuits contribute to learning. It also shows how tTIS can be used to study and potentially influence these processes. Imagine using this knowledge to help people who struggle with learning due to brain injuries or neurological disorders. This is an advanced critical appraisal discussion of the study in the spirit of evidence-based medicine. The first part is assessing the validity of the study. Let's look at the study design and methodology. This was a well-constructed, randomized, sham-controlled, double-blind study, which is a gold standard for investigating causal relationships. The inclusion of the sham stimulation as a control helps to mitigate placebo effects, and the blinding adds credibility. What about the sample size and what that says about generalizability? Well, the sample included 24 healthy participants, which is relatively small. While the results are compelling, the sample size limits the statistical power and generalizability. Larger, more diverse populations are going to be necessary in order to confirm these findings and extend them to clinical populations. What about internal validity? Well, the study design included precise targeting of the striatum using computational modeling to optimize the electrode montage or placement on the head. In terms of the frequency, the specific interventions of 80 Hz and 20 Hz, as well as the sham conditions, isolated the effects of different oscillatory modulations. However, potential confounders include individual variability in striatal structure and anatomy, and baseline oscillatory activity, which were not personalized in the protocol. What about analysis of the results? The key findings were that this study demonstrated that the 80 Hz tTIS disrupted reinforcement learning, while the 20 Hz stimulation and sham conditions did not. The disruption was specific to learning with reinforcement feedback, suggesting a causal role of high gamma oscillations in the striatum. What about the strength of the evidence? Their statistical analyses were robust, but the effect sizes, which were small to medium, highlight the need for caution when interpreting the clinical relevance. Cohen's D for key comparisons ranged from 0.2 to 0.5, which suggests moderate but not overwhelming behavioral effects. How about neuroimaging correlations? The results were supported by functional MRI data showing that 80 Hz tTIS selectively modulated neural activity in the putamen and caudate, key regions for reinforcement learning. This strengthens the causal link, but also raises questions about variability in neural response across individuals. How about applicability to clinical practice? What's the potential clinical impact? The findings suggest that tTIS could be a tool for investigating and potentially treating reinforcement learning deficits in neurological conditions such as Parkinson's disease or addiction. The ability to modulate deep brain structures non-invasively opens up new possibilities for therapeutic interventions. What about limitations in the clinical translation of this research? The study's findings were based on healthy young adults, so may not translate directly to older adults or clinical populations. Individual variability in striatal anatomy and oscillatory activity could limit the reproducibility of the effects in broader populations. What about practical barriers? Implementing tTIS clinically would require precision in electrode placement, which could be challenging in routine practice. Long-term safety and efficacy of repeated tTIS applications are also unknown. How about ethical considerations? The ability to non-invasively modulate deep brain regions does raise some ethical concerns about the potential misuse for cognitive enhancement or performance optimization in healthy individuals. What about access and equity? As with most cutting-edge technologies, ensuring equitable access might be a challenge, particularly if tTIS proves beneficial for conditions with significant socioeconomic barriers such as addiction treatment. What would be some recommendations for future research? First, larger and more diverse samples. It would be important to investigate the effects across broader age ranges and clinical populations in order to assess the generalizability. Second is personalization. It would be important to explore individualized protocols by tailoring the stimulation to each participant's specific neural architecture and oscillatory patterns. Third is long-term safety studies. It would be important to assess the impact of repeated tTIS sessions on neural integrity and overall brain health. The fourth is comparative effectiveness. It would be important to compare tTIS with other non-invasive brain stimulation techniques like RTMS or TDCS to evaluate relative advantages and limitations. The fifth would be integration with behavioral therapy. We could investigate tTIS as an adjunct to behavioral or cognitive training in conditions like stroke rehabilitation or obsessive compulsive disorders. This study represents an important step in exploring the potential of tTIS for understanding and modulating brain function. While these findings are promising, their clinical application remains speculative, underscoring the need for rigorous follow-up studies. Now that we know what tTIS can do, let's think bigger. How could this technology change the way that we treat neurological and psychological conditions? And, just as importantly, what challenges might we face as we move from the lab to the clinic? Let's start with the obvious question. How could tTIS potentially help people? Here are a few exciting possibilities. The first is in rehabilitation for stroke and brain injury. Imagine helping stroke survivors relearn motor skills faster by fine-tuning brain rhythms with tTIS. By targeting specific areas like the striatum, we could potentially boost recovery and help people regain independence. The second is in treating Parkinson's disease. Since the striatum is central to motor control, tTIS could be a non-invasive way to address some of the symptoms of Parkinson's, like tremors or difficulty with movement. The third is improving focus and learning. Beyond clinical conditions, tTIS might one day even be able to help healthy individuals improve their focus, memory, or even performance in skills like playing an instrument or mastering a sport. Now here's where it gets a bit tricky. With great power comes great responsibility, and tTIS raises some big questions. Even though tTIS is a non-invasive neurostimulation technique, we need to make sure that it's completely safe, especially with repeated use. Long-term effects need to be studied thoroughly. Should we limit this technology to medical use, or is it fair game for boosting performance in healthy people? What happens if it becomes a tool in high-pressure industries like sports or academia? How about access? Cutting-edge technologies like tTIS can be expensive, so how do we make sure that they're accessible to people who really need them and not just those who can afford them? Then there's the risk of misuse, whether it's unethical applications, overuse, or even just a lack of proper oversight. We'll need clear clinical treatment guidelines and regulations in order to prevent harm. So where do we go from here? The study that we're discussing today is really just the beginning. Researchers are already exploring ways to refine tTIS, making it even more targeted and potentially effective. Future studies might combine tTIS with other therapies like physical rehabilitation or cognitive training to maximize the benefits. There's also a lot to learn about how different frequencies of tTIS affect other parts of the brain. For example, could tTIS be used to treat anxiety, depression, or even addiction? These are all exciting possibilities on the horizon. tTIS has the potential to revolutionize neuroscience and medicine, but we're still in the early days. As this technology evolves, we need to stay curious, cautious, and committed to using it in ways that truly help people. Thank you so much for joining me today on the Neurostimulation Podcast. I hope that you enjoyed this exploration into the fascinating world of neuroscience and neurostimulation as much as I did. I really appreciate your time, your interest, and your attention. If you found this episode interesting, don't forget to like and subscribe to the podcast. It's the best way to make sure that you never miss an episode, and it helps us to reach more curious minds like yours. Also, if you think that this episode might resonate with a friend, a family member, or a colleague, please share it with them. Knowledge is better when it's shared, and you never know who might find this information helpful or inspiring. For more details about the research and technology that we discussed today, check out the links in the show notes below. You'll find everything that you need to dive deeper into the topic. And I'd love to hear your thoughts. Join the conversation in the comment section or reach out on social media. Your questions, ideas, and feedback make this podcast better. Finally, don't forget to tune in to the next episode. It's going to be another exciting journey into the cutting edge of neuroscience and neurostimulation. Thanks again for listening. Take care, and I'll see you next time on the Neurostimulation Podcast.